Hello, my name is Olivia Yochim, and my research focused on the role of estrogen receptor alpha on sex-dependent stress resilience. So a little bit of background on stress. It is a major risk factor for the development of major psychiatric disorders such as anxiety, depression, and post-traumatic stress disorder. Additionally, women are twice as likely than men to develop anxiety and depression. However, a bulk of stress neurobiology focuses on male rodent models. So our lab chose to specifically study the steroid hormone receptor mechanism of stress resilience. And so in order to do that, we studied the medial amygdala, which is high in both steroid hormone receptors, and the medial amygdala is a major brain region responsible for emotionality and stress response, mediating behaviors such as fear, aggressiveness, and defensive-ness. So our hypothesis was that estrogen receptor alpha expression in the posterior medial amygdala enhances resistance to social defeat stress in female but not male Syrian hamsters. And we chose the Syrian hamster model for two main reasons. One is that they display consistent territoriality and aggressive behaviors, and there's a similar pattern of aggression in both males and females. Some rodent models, the females do not tend to display as consistent aggressive behavior. So in order to study the effects similarly in males and females, Syrian hamsters were the best model for this. And we can also longitudinally see how their anxiety behaviors are changed over time by stress. So in order to test this, if you take a look at the bottom graphic shown in the white, this is sort of a timeline of our experiment. To begin, we conduct surgeries where we inject an adeno-associated virus into the medial amygdala, and the way that this works is a short hairpin RNA transcript will selectively bind to the RNA sequence for the estrogen receptor alpha RNA, and that'll prevent the translation of this estrogen receptor protein. So we have both a knockdown condition and a scrambled condition. In the scrambled condition, the virus does not prevent the translation of this estrogen receptor. So we have a condition where we knock down the presence of estrogen receptor alpha and a condition where it is not knocked down. Then the hamsters undergo something called a social avoidance test, where the hamster begins in a cage being acclimated to it, and then a novel hamster is introduced to its environment, and we measure how much time it spends interacting with the box with the new hamster in it. Then it undergoes social defeat stress by a larger, more aggressive hamster. This occurs 3 times for 5 minutes each, and then it undergoes social avoidance testing one more time. We measure how much time it spends interacting with that hamster after experiencing that social defeat stress from an aggressive hamster, and then we collect the brain tissue samples. Now, in order to determine whether the behavioral changes we're seeing are a direct result of the surgeries we performed, if you take a look at the graphic with the green and the red fluorescence, we use immunohistochemistry and fluorescent microscopy in order to, one, determine if the injection successfully targeted the medial amygdala, and 2, to see if estrogen receptors were successfully knocked down. So in the green condition, the fluorescence demonstrates the presence of the virus, and we can see that it successfully targeted the medial amygdala, and then in the red, that shows all the cells with estrogen receptor alpha, and as we can see in the medial amygdala in the D image, it shows that there's a lot less of those red dots, showing that that virus was successful in knocking down the estrogen receptor alpha. And so if you take a look at our results, so far what we're finding is that in the scrambled female condition, the females are not displaying a change in the amount of time they're spending interacting with that novel hamster, both before and after experiencing that social stress. However, if we take a look at the knockdown females, shown in the orange, we can see that prior to being exposed to a social defeat stress, they are spending more time interacting with the hamsters than the scrambled or with their post-defeat results. So after being subjected to stress, they are now spending less time interacting with the novel hamster box. And this is kind of a similar pattern of behavior that we're seeing in the males. However, both the scrambled and the knockdown male hamsters show a similar trend of interacting with the box before being socially stressed and then interacting with it less after being socially stressed. So to sort of wrap things up, a discussion on what is the impact of estrogen receptor alpha, we can say that males tend to demonstrate more stress-induced anxiety than females. However, the females with the knockdown of estrogen receptor alpha show heightened anxiety-like behavior following that social defeat exposure, and it's similar to the male-like pattern of stress-induced anxiety. So a question that we had is estrogen receptor alpha really playing a role in stress resilience? Because unexpectedly, the activity of ER alpha may reduce the hamster's pro-social behavior and thereby modulate stress resilience. We said this because in the knockdown female condition, those hamsters were more willing to explore than both the scrambled and their post-stress condition, and it was really the fact that they were interacting with the other hamsters more that resulted in the big changes that we're seeing in the data. And ongoing histology is needed to confirm the accuracy of our injections, of our surgeries on the behavioral results that we're seeing. But I just wanted to thank the Cooper Lab, specifically Dr. Matthew Cooper, for supporting me in this research, as well as the graduate students and undergraduate students in that lab, the NIH grant that funds our research, and the advanced undergraduate research activity that has funded my role in this research project. But thank you so much for listening, and have a good one. Bye-bye.